Hi, my name is Graham Dick. I am the product manager for BioIVT's ADME product and services group. Before I introduce Brian Ogilvie, our speaker on today's webinar, I would like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to be the largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the biospecimens and services our customers need to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and be the preferred provider to our global biopharmaceutical clients. And because Brian is legacy Xenotech, I do want to highlight the Xenotech acquisition in 2022 that significantly expanded our ADME research services capabilities. We have invested in sourcing products globally and an extensive global distribution. We stage products in regions where our customers are located. This helps to ensure short turnaround times and enable us to get products to our customers' labs when they need them. Underpinning all of what we do is our commitment to ethical sourcing, quality, and a sincere desire to work as a partner to the research community. Here we see a snapshot of the array of BioIVT's portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and products and services to evaluate the ADME properties of new drugs. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts routinely works with clients to help them identify hepatocyte lots with the characteristics that meet their specific requirements. Additionally, we help clients design and implement their preclinical ADME research programs and develop data packages for IND submissions. Our fit-for-purpose designs include programs in drug metabolism, enzyme induction and inhibition, drug transporter, and other ADME tox research services. Visit bioivt.com to learn more about how our portfolio of products and services can meet your needs. It is my pleasure to introduce my colleague, Dr. Brian Ogilvie. Brian earned his PhD in toxicology from the University of Kansas Medical Center. In 1997, he joined Xenotech, and over the last, now more than 27 years, has been key in developing Xenotech's reputation as a company that is truly dedicated to designing and implementing in vitro ADME programs that achieve clients' objectives, whether that's proof of concept, lead optimization, in-licensing or out-licensing assets, or taking drug candidates through an IND submission and clinical development. In 2022, when Xenotech joined BioIVT, Brian assumed his current position of Vice President of Scientific Consulting. In this role, he is part of our team that advises clients on their preclinical strategies, always taking the perspective of understanding their R&D goals. When you registered for the webinar, we asked you to respond to questions about how you expect the new guidance to impact your drug development programs. I think the key takeaway from these poll results is that about half of you are concerned about how the guidance impacts past and current studies and regulatory submissions. Also, over half think their current practices do not meet the guidance or are unsure. Brian, I think these results show your presentation is very timely. We look forward to hearing your thoughts.
Yeah, I appreciate that. And also on the outsourcing question, it looked like about 1/3 completely outsource and 16% are completely in-house. So I'm hoping this presentation will give you some pointers from the final ICH guidance that you can use when selecting either outsourcing partners or designing your own studies. And I would also mention that in my consulting role, this is something that I help people with quite a bit. So I'm going to share my screen and then make sure I've got that up. I'm going to turn off my camera just to make sure I have enough bandwidth going forward. So let me go ahead and do that. And I should be ready to go. So as Graham mentioned, this is the title of the presentation for the final ICH M12. And if you look at that presentation outline, in the interest of time, I will be focusing only on the in vitro sections of the ICH M12. There are actually some new clinical developments, such as the biomarker section, that I think many people are interested in. That could probably be its own separate presentation that I won't get into today. And if you look at the current regulatory landscape, and I say current because the final ICH M12 is technically pending adoption by all the agencies. So looking back though, these guidance documents may be currently in effect for a while, but going back to 1997 when FDA and EMA first published drug interaction guidance, the final ICH M12 really is then the culmination of 27 years of scientific and regulatory progress in drug-drug interactions. And as Graham mentioned also about the exact length of my career so far, and it was a privilege to contribute to the science along the way, some of which has actually entered the guidance documents. So moving on, the final ICH M12 is really one guidance to guide them all. And I say that because you still have agency-specific interactions. You also have the regional documents that are in effect for some areas. And nuances can vary even between reviewers at the agency. So it's really important to know your audience. You can't just go in there with the ICH guidance and say, hey, this is final, this is exactly what it says. There are specific instances in which a reviewer may ask for something slightly different. And again, like I mentioned, the regional guidance on specific topics still apply. So for example, oligonucleotides, ADCs, oral contraceptive steroids, et cetera. And if you look also at FDA, one of my colleagues actually spoke to someone from FDA in May about the effective date. And they said that essentially the ICH M12 was already in effect as of the 21st of May, 2024, and replaces the FDA guidance. However, there's no formal adoption notice in the Federal Register yet. And if you look back to the draft ICH M12, it was about 3 months later before that entered the Federal Register, and FDA put it into their own format, signaling that they have now adopted that draft guidance. The person at FDA, again, this is a personal interpretation, not a view of the agency as a whole, said that for a little while, if the question about ICH M12 isn't directly asked and data were clearly generated a while ago, some review teams would probably approve them as long as they have good quality safety data. However, if you do go and directly ask about the ACH, the response would probably be that the M12 should be adhered to. So bottom line really is that you should review the current and planned in vitro DDI studies that you have with the final ICH M12 in mind now. And would that change the interpretation? Or are there additional experimental considerations to meet the final guidelines recommendations? And in addition to the slides, I've got links here that you'll have in the slides for the actual final guidance document, the Q&A document, which is important, as well as the training presentation. Now, I did find out recently that EMA added an official copy to their website already in late June, about 1 month after officially adopting the ICH guidance document. And interestingly, now they've put this date for coming into effect at the end of November. So now we know EMA is not going to consider the ICH M12 final until the end of November this year. But on the website where they post this, there's a little bit of a curveball. EMA states that of particular note, ICH M12, unlike the existing EMA guideline, does not address drug interactions in the gastrointestinal tract to the same extent. And this will be addressed by the development of accompanying EMA guidance anticipated to take the form of questions and answers document. So we don't know what this looks like yet. I'm kind of wondering if it's in terms of the equation for time dependent of CYP3A4 in the intestine or also transporters. By the way, it certainly appears that EMA will have somewhat different requirements for DDIs in the gastrointestinal tract. So that's interesting to note. And you also find one of the conventions I use throughout this presentation is that when I'm quoting guidance, I typically quote that. I try not to paraphrase, and in some cases I will actually paraphrase, but it's not something that I want to do lightly. For the most part, I'm going to go through some notable changes in the final versus the draft guidance that came out 2 years ago. And for the most part, they're not sweeping changes from the draft to the final, but it's certainly clear that over the 1,000 comments that were received, they were read and many were addressed either in the guideline directly or in the Q&A document or even the training presentation, which I linked earlier. So my convention will be that the text I underline in a passage means that part is new and I considered it to be notable. Others are just new or changed to a minor extent. So getting into it, nomenclature, this is actually the #1 comment. There was only, I should say the first comment, there was only one comment along this line that the use of victim and perpetrator may be sort of outdated. And ICH did take that into account, and they're now replacing those terms with object and precipitant, which is a common term that has been used. If you look back at the University of Washington drug-drug interaction database, which is now owned by Certara, they have used object and precipitant for years. And then for plasma protein binding, there's a new section actually that describes when a measured fraction bound in plasma can be used for all drugs, including highly protein bound drugs. So in other words, when you have greater than 99% binding. So, and this actually removes the old limitation of no less than 1% free in your extrapolations, even if it's 99% bound. So this can make the basic model and other conclusions less conservative than before, as long as the accuracy and precision of the protein binding measurement has been demonstrated. Such a demonstration actually should include validation data of the protein binding, including bioanalytical method, and an appropriate positive controls. In other words, drugs with high binding to relevant plasma proteins. So I think most people have the ability to do this, and that's good news for most of us that have highly protein bound drugs. On the timing, they did change a few things based on comments. Previously, it looked like, and I commented this in previous webinars as well, it said reaction phenotyping should be performed before phase 1. So, most of my clients actually do a little bit of reaction phenotyping as IND enabling studies. But for those who want to wait a little bit, now you can move it into phase 1 in patients. So that would be, in other words, in something like oncology, or it can wait till phase 2 when you're actually in patients if you're using healthy volunteers in phase 1. So a little bit more leeway there. And then for CYP and transporter inhibition, CYP induction before said, before initiating larger studies in patients. So that's kind of up for interpretation. If you have a relatively large phase 2, that might mean before that. Potentially, if you have very small phase 2 studies, that could mean prior to phase 3. So again, providing a little bit more leeway in terms of timing. But again, most of my clients will actually do CYP and transporter inhibition, certainly as IND enabling studies. And then when it comes to human mass balance studies, this was actually addressed directly in the Q&A document because of the comments. Now ICH is saying clinical DDI studies can be conducted based on information from in vitro studies prior to obtaining additional information from mass balance study. And then paraphrasing a little bit here, we do not, and then direct quote again, intend to restrict the timing of a mass balance study for DDI evaluation, and that flexibility should be ensured according to the characteristics of the investigational drug. Of course, mass balance data are very important in making some of these decisions, but it's not always practical to have those early on. And then when it goes to CYP induction, it's a little bit of a mixed bag, to be honest. So if you look at the basic mRNA fold change method, they have 2 criteria. Clinical induction can't be ruled out if the drug and hepatocytes from any donor meets both of these criteria. Increases in mRNA expression of a CYP enzyme in a concentration-dependent manner, and the fold change of CYP mRNA is greater than 2-fold at concentrations less than 50 times the unbound Cmax. So this is a change from the draft guidance where it said greater than or equal to 2-fold at 15 times the unbound Cmax. And it's also a change in the appendix that now the concentration should encompass 50 times the unbound Cmax, not 15x as in the draft. They also have additional wording on having positive controls greater than 6-fold, and they note that if the positive control has less than 6-fold, the induction potential cannot be ruled out. If that drug increases CYP mRNA less than 2-fold, but is more than 20% of the response to the positive control. So a little more prescriptive there, and also put some more impetus on having positive controls in hepatocyte lots that will reach more than 6-fold increase in mRNA. They do call out that it's not mandatory for CYP2Cs. I think the science is pretty developed on that. And they have additional clarification. Previously, they only said that CYP2Cs needed to be evaluation if CYP3A4 was induced in vitro. Now it says if any clinical study also shows that the drug candidate induces CYP3A4, you should go back and look at the CYP2Cs, either in vitro or clinically. But on the other hand, now the equations list the EC50 unbound throughout. So this would require determination of unbound concentration in hepatocytes. Some people may have already been doing this, but it may require some work for others. And the methods cited are based on a 2017 and a xenobiotic, both drug metabolism disposition and xenobiotic papers from Sun et al. And Cheng et al. So those references are provided in the guidance, and more induction examples are actually provided in the Q&A document, which are quite informative. Okay, so other changes from the draft. It just minor clarification that as a substrate of drug metabolizing enzymes, they clarified that CYP3A means both CYP3A4 and CYP3A5. And then on P450 time-dependent inhibition, they do clarify that non-dilution IC50 shift assays should be used. So this was nice to see by many commenters, cited our 2011 paper on using the dilution method. I would note that the ICH did not actually cite our paper in the guidance, but they do allow for non-dilution methods for the IC50 shift assays to be used now. Also on the UGT inhibition, that list was included to expanded to include not only UGT 1A1 and 2B7, but also 1A4, 1A9, and 2B15. And you'll note that this list is quite a bit shorter than what's listed in the reaction phenotyping section, but I believe this is based on the idea that if direct glucuronidation is the major elimination pathway of a drug, then you should look at these. And the list seems to be based on two John Miner's publications from 2021 and 2023 that the ICH cites. For transporter inhibition, they actually removed the list of other transporters. I will note that it is included in the substrate potential for transporters, but now we no longer see BCEP. Actually, BCEP is not mentioned anywhere in the guideline, the final guideline. MRP2 are also removed, and it recommends that the decision to evaluate additional transporters can take into consideration if commonly co-administered drugs or substrates for other transporters. They don't actually list what those other transporters are. It's sort of up to you to decide if you may have an interaction there and test that. The other sort of big change here is that now IC50 unbound is listed throughout, requiring determination of unbound concentration in any in vitro system that's used for transporter inhibition. And then I want to comment on these slides in particular are really references. They've been very popular in similar webinars, so I won't go through all of them in detail, but they can be quite useful for evaluating whether or not your data interpretation would change. So if you've been submitting to PMDA or EMA, for instance, you can look at some of the equations later on, see what the ICH is now recommending. Did your interpretation change based on that? Also, because you'll receive copies of these slides afterwards, you'll have these as a reference. And my convention throughout is that the red text is when the ICH was different from other guidance. The bold red is a change from the draft guidance. So you can see for this, evaluating drugs as objects according to the different guidance documents, it's all the same, with the call out that ICH, as I mentioned, has this longer list of UGTs and also has added the N-acetyltransferases. But again, there's no change from the draft in that regard. Now, when it comes to evaluating test drugs as transporter substrates, in other words, objects, there's really not much change here. I will call out that they did change the phrasing for BCRP and PGP to say that they're often evaluated. In other words, not always. They do refer actually at one point to the ICH M9 for the biowaiver guidance. And then they changed some minor wording. They also note that for hepatic uptake, you should also look at that beyond biliary secretion greater than 25%, but also if the pharmacological target is in the liver. And then of course for renal, if active renal secretion is greater than 25% of systemic clearance. I will note that the ICH final document did seem to remove the renal toxicity observed requirement that was in the draft. At least I could not find it. I did an extensive search for that. And then for the other transporters, again, they've removed BCEP entirely, MRP2, OCT1, and OATP 2B1. They note just minor wording changes here, really. Additional transporters may be considered on a case-by-case basis as new information emerges and our understanding evolves. So just recognition that guidance is relatively static. I mean, we may have a revision for this guidance later on. We know that EMA is going to produce a sort of ancillary guidance or Q&A document, but science marches on and our understanding will too, and so it's just an acknowledgement of that fact. ICH also has this, and this is no change from the draft, but for PGP and BCRP if biliary excretion as a major pathway, and PGP if active renal secretion is a major elimination pathway. And then a simplified interpretation of the substrate potential for transporters. So essentially the same as the draft. Again, it notes that other cutoff values or relative ratios to positive controls can be used if prior experience justifies alternatives, and sponsors can certainly propose criteria for vesicle-based systems based on prior experience. But essentially, these are all the same as before. The ICH also does have additional considerations in the appendices mentioning MRP2, OCT1, and OATP2B1. They also note that passive permeability must be considered, and you must compare test compound to a high permeable control, and you need saturating concentrations or use transporter inhibitors if validated high and low permeability reference standards are included. And then again, it refers to the ICH M9 BioWaiver Guideline. And then for transporters that are expressed in multiple organs, PGP and BCRP, they now say that sponsors should provide justification for the choice of substrate concentration. And if the in vitro system expresses multiple transporters, so for example, CACO2 cells, you may have to conduct additional experiments to confirm findings with 2 or more known inhibitors. And again, those appear in the appendices. That would probably be its own webinar on its own. So I'd be kind of curious if anyone wants additional webinars that go into the nitty-gritty details that are in the appendices. And then moving on to drug metabolizing enzyme inhibition as a precipitant, the final ICH has added the 3 more UGTs, as I mentioned here, if direct glucuronidation occurs with your drug or co-administered drugs, including 1A1, UGT 1A4, 1A9, 2B7, and 215. And they did change minor wording when an investigational drug will be commonly administered with a drug. And I think also you might consider looking at fixed dose combinations when you know that the other drug will be glucuronidated. So an interpretation of reversible hepatic CYP inhibition. There's really no change from the draft except to mention that this equation for ICH, the Cmax unbound over the Ki unbound with the same cutoff value of greater than 0.02 may be able to be applied to UGT inhibition. They do note that it's an area of ongoing research and that it may not apply in all cases. They do also cite our 2015 paper showing that you can calculate Ki values as IC50 over 2. So in other words, just because they put Ki unbound in the equation, you can go ahead and determine IC50 values instead of just Ki values all the time. When it comes to reversible inhibition of intestinal CYP3 enzymes, the final version did fix the lack of unbound in vitro Ki. So that's now listed in the equation. I've highlighted it here. And also I would note that the ICH equation can be written as when you have Ki unbound greater than 0.1 times the maximum clinical dose in 250, that cutoff value would still be 10. These are considered no effect boundaries. I'm not going to cover the mechanistic static models in great detail. You'll notice that I'm focusing mainly on the basic models. And when it comes to irreversible inhibition of hepatic CYP enzymes, there is essentially no change here. ICH does have additional experimental conditions or considerations that are detailed in the appendices, but you can see that as before, the ICH draft lowered that 50-fold down to a 5-fold safety factor because of the overprediction that was occurring in the literature that was showing that using that 50-fold safety factor for the irreversible inhibition. And when it comes to irreversible inhibition of intestinal CYP3A, again, as with the draft, there still isn't an equation for that, as there wasn't for FDA 2020. So this was a little bit of a mystery to me before 2022. I heard at the EU DMDG meeting in Amsterdam, there were some EU regulators there that explained that this equation grossly overestimated time-dependent inhibition in the gut, and that the new hepatic equation with the 5-fold safety factor covers it. But I do wonder if EMA's comment about addressing drug interactions in the gastrointestinal tract has anything to do with this, or if it's only related to transporters. So we'll definitely be watching for that. And then moving on to induction, again, the only thing that, well, there is some major change, as I mentioned previously. Now we have to determine EC50 unbound. So that means determining the unbound concentration in hepatocytes. There are also additional details in the appendix, but basically the cutoffs are all the same. There is one thing I want to highlight. So when it comes to looking at the spent media analysis from induction studies, they now call out an 80% threshold. So in other words, if you have less than 80% left at the end of the incubation, sponsors are encouraged to measure the concentration of parent drug in the medium on the last day of the incubation. And when that's lower than 80%, due to non-metabolism transporter-related conditions, sponsors should discuss the potential impact of the discrepancy on the data interpretation with regulatory authorities. So in previous guidance, it just told you to look at the spent media analysis. Now it tells you what to do and puts a threshold in there of less than 80%. And the Q&A document also suggests some consideration of instability, and that correction for instability or more frequent refreshment of the medium should be considered in those cases. There are additional considerations mentioned for UGT induction and potentially even transporter induction, but not very much specific guidance there. Downregulation has been mentioned in the previous guidance as well, with less than 50% of the control mRNA warranting additional in vitro or clinical studies to understand further. So transporter inhibition now. The ICH did change a few things. It's the same list as FDA and PMDA below. They now have removed for transporter inhibition the list of other transporters and simply say, as I mentioned before, sponsors can consider evaluating the inhibition potential of a drug and other transporters as necessary. So the decision to evaluate that needs to then take into consideration commonly co-administered drugs. And as literature and science move on, if you know that there may be a potential interaction with a certain transporter, that would be a call to potentially look at that in those cases. And then time-dependent inhibition of other transporters, particularly OATPs, but others, that literature is pretty mature now. So the ICH leaves in there to follow the current literature, and then they have additional details in the appendices. And for transporter inhibition and substrate potential, those really could be, as I mentioned, another presentation. So moving on then to intestinal PGP and BCRP inhibition, the final version did seem to take the comment that Ki values aren't needed for transporter inhibition, especially for transporter assays that are typically conducted at substrate concentrations very much lower than Km. And you'll see that reflected here. The Ki value is deleted from those equations and it's only IC50, but it is unbound. And I should also mention that the cutoff values are listed for orally administered drugs for PGP and BCRP. So for other routes, IV, or for metabolites that are formed post-absorption, the unbound IC50 greater than 50 times Cmax can be used with a cutoff value then of 0.02. And as before, they do note that other cutoff values can be proposed if justified based on an in vitro to in vivo extrapolation and calibration of specific in vitro systems. There are many papers published on this topic, and I know a lot of companies that actually use alternative cutoff values based on the literature and the publications that they have spearheaded themselves. So for the OATPs, the final version, there's really no change here. The final version did, in fact, include the blood to plasma ratio in the calculation of the Cmax inlet unbound. So that can now be used. And for discussion of that blood to plasma ratio, I would encourage you to look at a letter to the editor from Andrew Parkinson to Drug Metabolism Disposition from 2019, where he discusses the differences between the PMDA and the other guidance documents in terms of using that blood to plasma ratio and calculating the Cmax inlet concentration. And essentially there's no effect based on the way the equations are written. But it is important to know that you do need to have that blood to plasma ratio, which can of course easily be experimentally determined along with plasma protein binding. So moving on to the OATs, OCT2 and mates for inhibition, the equations and cutoffs. There's essentially no change here except the fact that again, they've removed Ki and they've included unbound IC50. So I've highlighted that here. And the mates remain at the more conservative EMA and PMDA cutoff of greater than 0.02. So this is one that went back and forth a little bit with FDA finally setting on 0.1. And as I mentioned a couple years ago, when the draft ICH came out, they did go back to the 0.02. The ICH does not explicitly cover MRP2, OCT1, or OATP2B1 or other transporters in the cutoff equations, so it sort of leaves what those cutoff values should be, and if they can potentially use any literature that's come out to justify those cutoff values for any other transporters that would be looked at for inhibition. So moving on to drug-drug interaction with metabolites. In the final version, there's really only minor clarifications or wording changes as metabolites for substrates. It also notes, as before, generally not needed if there will be a clinical DDI study of the parent. So if you know kind of early on, you see really strong indication that you're going to be conducting a clinical drug-drug interaction, you probably don't need to evaluate the metabolites in vitro. When it comes to metabolites as substrates, again, the minor change here really is just that if on-target effect of a metabolite is greater than the parent, and everything else is really pretty much the same. It does differ a little bit from the 2020 FDA guidance for metabolites saying specifically greater than 50% of overall activity. And then there are additional details in the guidelines that I won't belabor here. For inhibitors, it's certainly more clear now that it's referring to human mass balance studies. And this was a comment that I made in webinars in the past and that many commenters submitted in writing, when do we have to have the mass balance studies? So they are saying that for metabolites, you have to have that greater than 25% of AUC apparent and at least 10% of drug-related material. And they now say often determined based on radioactivity data. However, elsewhere in the guidance, they do clarify that looking at metabolites as inhibitors and so on can be conducted later in your development program. However, most of my customers and clients actually do look at metabolites as inhibitors if they're major based on early data, not necessarily mass balance data. We'll go ahead and put those into their early, possibly even IND enabling studies. And they also add this clarifying statement here in the final ICH guidance that usually metabolites are irrelevant for evaluation of intestinal CYP or transporters except when formed substantially in the gut or intestinal cells. So I think that would be a little bit difficult to know unless you have strong data from later studies showing that you have metabolism in the gut or in the enterocytes themselves. There are some other minor clarifications here. Similar approaches are used for transporters and P450s. And then generally no assessment of metabolites as inducers. They do have previously, unless a prodrug or a major metabolites form extrahepatically, and now they add in the final and at least 10% of drug-related material. So again, referring to potentially mass balance. And then more conditions or considerations from the appendices, experimental details. There's really no time to cover all these changes. Most that haven't already been called out are pretty minor. They do have this new plasma protein binding section, which includes recommendations for the bioanalytical validation and validation with very highly protein-bound compounds. They do have more complete lists now for drugs that can be used in studies of CYPs, UGTs, and transporters, and even on the clinical side as well as, and then the predictive PBPK modeling. I won't belabor that right now. Most companies obviously do want to do modeling at some point, and I would direct you to the separate guidance for PBPK submissions. When it comes to test systems, previously they said for human liver microsomes, a pool of at least 10 donors suggested, but now there's this change for UGTs. They now require that if human liver microsomes are used for UGT studies, that it requires either alomethicin or sonication and always to use bovine serum albumin. And the reason for this is just kind of to put the UGT activity all on a level playing field across all sponsors. And then and I say that because the active site for UGTs is on the lumen of the endoplasmic reticulum. It's a little bit of an argument with human liver microsomes. Some of them may be inside out, so it doesn't make much difference. But either way, it's now in the final guidance. And then BSA minimizes inhibition by long chain fatty acids, which is well known for some UGTs. And then it lists the other systems as before and which enzymes may be present there. And then for hepatocytes, this was interesting to me because before they said that for phenotyping and inhibition, hepatocytes pooled from at least 10 donors is suggested. However, there was a comment that said, well, why do we have to have n equals 10 when it's only n equals 3 for induction? To me, that's comparing apples to oranges, but they actually seem to take that into consideration and lowered that to 5 to 10 donors. And that may be important for some people that have specific lots in-house that they are using and have selected. But from my perspective, I was thinking, BioIVT, we already offer pools of up to 200. And when you're down to a pool of 5 hepatocyte donors, you have to start worrying about polymorphisms, especially for these phenotyping experiments. So something to pay attention. I would always recommend using a high number donor pool. They do also still have the requirement that for induction experiments, if you have a single culture that's fully, previously said validated, now they say fully qualified per the ICH M12. The only downside to that is that you can qualify that according to what they say, but if you run out of that lot, you're going to be doing that whole qualification again. And as before, for induction, mRNA is the endpoint, except for 2C19, where activity should be used. And then the test article concentration, as I mentioned before, is now up to, it should encompass 50 times the unbound Cmax, not 15 times as the draft. So it's a little bit of a, I think, trying to not be as conservative with the unbound EC50 being used by adding that. I don't know exactly what the deliberations were, but that's where they settled for the final guidance document. So for therapeutic protein DDIs, I'm not going to belabor this. There's just very minor clarifications in the text. Previously, I had noted that proteins are not peptides, and ICH now specifically notes that oligonucleotides, small interfering ribonucleic acid, modified ribose nucleic acids, and peptides are out of scope of the M12 guidance. And I would mention that, again, there are regional guidance documents that refer to many of these. And then on pro-inflammatory cytokine-related mechanisms, there's essentially no change from the draft. There are other minimal guidance or details provided in the guidance. The only thing I would call out here is that we do have patented methods at BioIVT to investigate these effects for both cytokines or immunomodulators, whether they're small or large molecules. And then moving on to antibody drug conjugates, I'm including this only for completeness. There's essentially no change, some minor clarifications. I would note that they say that for a small molecule component, the potential to inhibit or induce enzymes and transporters should be addressed in a similar manner as described for small molecules in the guideline. And possibly you might have to look at the small molecule component as a substrate. I will say we have been running these studies for at least 20 years, going back to some of the first ADCs that were developed. And now that that field is pretty mature, there are well-characterized payloads. And in many cases, I've seen companies that take strategies where they're using well-characterized payloads and relying on previous data that was generated for the small molecule and linker component for those. So to conclude, we're almost there. We will be there by the end of the year. EMA plans to issue an accompanying Q&A document, as I mentioned. We'll be quite interested in seeing what's in there. They will adopt the rest of the guideline by the end of November 2024, and I expect that with the FDA's stance that they have essentially already adopted it. It's just not in the Federal Register yet in their format, but once that occurs, probably in August, we can consider that formally adopted by FDA. Many details in the ICH are still identical to previous guidance documents for the in vitro DDI at least. And the specific regional guidance are still in effect. So oligonucleotides, ADCs, oral contraceptive steroids, and others. And one thing I would call to your attention, the appendices now can be used to review completed studies since this is final for the quality of methods used, and to review, bottom line, basically review current and planned in vitro DDI studies with the final ICH M12 in mind. And with those equations that I have in this document, would it change interpretation? In other words, there's a little bit more conservative on induction. Is there a case where now maybe you're over the threshold for that and might have to consider a clinical DDI study? Or are there additional experimental conditions that might need to be conducted to meet the final guidelines recommendation? And the guidance realizes that science evolves. It leaves it open for new approaches. And in spite of harmonization and guidance, remember that each agency or reviewer or individual office within the FDA, for instance, may interpret aspects differently. So be sure you know your audience when you're going to submit, and feel free to take the opportunities to communicate with the agencies. FDA has opened up more doors for that to occur early on. And also we can, and I'm quite used to doing consulting projects, looking at legacy data or even plans for new studies and determining whether or not it will meet the current guidance and whether or not there will be questions from FDA or EMA on particular data that's available. So with that, I'll take any questions or comments. I see some in the box, and I think got Graham back on board.
Yeah, thank you, Brian, for that great presentation. Yeah, we just want to remind everyone to submit any questions in the question box or the question pane, and appreciate the questions that are in here so far. So I'll read the questions, Brian, and then get you to respond. First from Raj. While determining the DDI potential for reversible and time-dependent inhibitions, unbound Ki and Kl are used in sections 2.1. Well, I won't go on. However, in the section on the mechanistic status model, section 7.5, the terms Ag, Bg, Cg, AH, BH, and CH do not mention unbound Ki or Kl, and just mention Ki. Is it just a typo, or are we required to use unbound Ki and Kl in all cases?
That's actually a good question. I did notice that, and like I said, I didn't focus on the mechanistic static models. I think that is a carryover from the way the equation was written before, but I think with using the unbound Cmax inlet, which is described in the footnotes for those equations, I think it is covered if I remember correctly. So I believe, given that the mechanistic static model is a little bit less conservative, it would seem appropriate to still use the unbound in vitro concentrations to me.
Okay, thank you, Brian. From Rotash, do we need inhibition studies with a circulating N or O glucuronide metabolites with AUC greater than 25% parent AUC in human ADME study?
I would typically say yes, and I think this goes back to my experience from like 2006 through 2008 when we finally published on this, that gimfibrillol acyl glucuronide is a potent mechanism-based inhibitor, CYP2C8, and actually is the mechanism for why cerevistatin was withdrawn from the market when it was co-administered with gimfibrillol. So definitely glucuronides, even if they're O or N glucuronides, have the potential to inhibit P450s or actually UGTs in some cases, so or transporters for that matter. And so I would definitely, if those metabolites are major, need to be evaluated, in my opinion.
Thank you. From Peter, since mRNA is the endpoint for CYP induction, would you suggest not doing activity for CYP1A2, 2B6, and 3A4 at all?
Yeah, I think that comes down to personal preference, but in my opinion, if you have, so the reason that came about, going back to Pfizer papers, I think were published maybe around the 2007-2008 timeline, and the FDA changed their guidance from being relying on activity to mRNA. The reason for that was because if you have mechanism-based inhibition, time-dependent inhibition rather, of any of those CYPs, then your activity would be artificially decreased. So that makes mRNA the better endpoint. So I think to simplify things, I would personally just focus on mRNA as the endpoint, except for 2C19. And there are some cases actually where some of the other CYP2Cs don't have as robust of an induction with mRNA, and you may actually have to look at activity there as well.
Thank you. And from Grace, for CYP induction, if one of the donor showed positive signals in mRNA, can you de-risk it by showing activity data?
That's a good question. I don't think the guidance specifically said that, but again, because it allows for some leeway in terms of scientific justification. I think if you can make a good case for that, that's a possibility. But again, you would have to also show that there is no time-dependent inhibition that's confounding that. In other words, yeah, you saw an mRNA signal and the activity looks clean, but is that because of inhibition of that enzyme and activity there? So I think there's some big caveats to taking that approach.
And I think a related question from Grace at BioIVT, do you usually advise clients to run both mRNA and activity for CYP induction?
No, we typically focus on mRNA, again, except for CYP2C19 in particular. And we've also looked at activity in many cases for the other CYP2Cs. We do also have some clients that just have a preference to run both all the time. And that comes down to a question of, do you want to have, are you a little bit more risk averse? You want to just be more thorough in looking at both endpoints, and that's certainly fine. And we don't typically see a large difference there unless, again, there is some time-dependent inhibition.
And I think a follow-up here from Peter, for NDA submissions in 2025 and beyond, but for studies performed before in 24, which guidance should be used in your opinion?
Yeah, so that comes back to that conversation my colleague had with FDA personnel. And again, anytime you're talking to someone from FDA, that's not the official position of the agency. But I think they were saying that certain reviewers would probably accept older data as long as it was high quality. But the official stance would be that the ICH M12 is in effect. However, we've been through this before. So going back to 2017, there was one instance where we had a company that even prior to the draft 2017 FDA guidance came to us in a panic and said, look, FDA is putting some restrictions on our phase 2 study because we don't have any transporter inhibition for all these others besides PGP and BCRP. And then those transporters appeared in 2017 guidelines. So they were already looking at that. So I think it's possible that there will be some reviewers that will take a conservative stance and say, yeah, you do need to comply with the ICH M12 in all aspects. Again, given the relatively minor differences in some cases, I don't know there's going to be a large impact in some of those cases. But like I said, I'm used to looking at legacy data and going through and trying to formulate the approach and the strategy that should be taken going forward, and even being able to correspond with the agency and come up with a good compromise. Some of those possibly might be post-marketing requirements or commitments.
Thank you, Brian. From Kelvin, how do we deal with compounds that have limited solubility that would not meet the 50 times Cmax?
Yeah, the guidance certainly addresses that. And then also in terms of cytotoxicity, if you're talking about hepatocytes or any of the cell lines that are used for transporter inhibition or substrate potential. And of course, they recognize that in certain circumstances, you just simply can't reach those concentrations. And you just have to justify that. And in cases where you could potentially have a false negative because of that, then that's something you have to discuss with the agency and look for a way forward. But I mean, they don't expect us to, change the physics or try to really, you certainly can change solvents to some extent, but then you're worried about inhibiting some of these enzymes. We've had companies that go as high as 0.2% DMSO, and we know those solvents will inhibit P450s. So it comes down to compromising and finding out what you can actually do, practically speaking.
Thank you. From Olivier, can you comment on the acceptance of the M12 guidance by PMDA, so the Japan agency, and CDE, China agency?
No, I looked, and it's all in Japanese, and I couldn't get a good translation, so I couldn't see anything on the website that showed any mention of ICH at this point. So I may reach out to a Japanese colleague and see if they can look for me, but I have not found that out yet. That was one question that certainly occurred to me.
Thank you. From Juan, with the new request to determine unbound concentrations for transport inhibition, are there available assays for FU, INC with vesicles? And if we use recombinant cells, should we test FU, INC with all of them expressing different transporters, CHO, FDCK, and so on?
Yeah, that was our take, that we're going to have to look in each different cell line, and there's a variety of different ways of doing that. I don't want to comment specifically on how that would be conducted. But it can certainly be done. There may be surrogate endpoints that can be used to come up with that. For vesicles, I know that they're a little bit more restrictive on vesicles in the appendix. I didn't cover that to a great extent, but essentially, I think for the mates, you'd have to provide for vesicles additional justification for using that test system. And I don't know what the best way is, honestly, to look at unbound concentration with vesicles. I think you could do something along the lines of, simply with and without the control vesicles, and determine if there's binding to the apparatus versus binding to the vesicles themselves.
From Jasleen, how do you recommend measuring fraction unbound values for transporter hepatocyte-based studies? Microsome binding as a surrogate, for example?
I wouldn't use microsome binding as a surrogate for transporter binding because they're different cell lines. I think that there are ways and ways to do this, and there have been some literature reports on how to look at that. We're actually working through that right now, and I don't want to spill the beans, so to say, on how we're going to approach that, but there are multiple approaches. So I hate to be a little bit cagey on that one, but I don't want to speak too soon on that.
Zipping along, trying to get through these before the end of the hour. From Jing, can you speak about the clinical approach to testing the UGT inhibition? And Jing, we also thank you for your compliment.
Yeah, for clinical, to be honest, I did not focus on that in this presentation. I've certainly read through it. They do include some of the substrates that can be used clinically. But again, I think that's probably something that's still developing. I would need to go back and take a closer look, to be honest, at the clinical section.
Okay. From Mark, is there specific comments in the appendices on conducting DDI studies on PROTACs?
On PROTACs, I did not see mention of that. They may have used a different phrase than PROTACs, but yeah, that's certainly something that more and more companies are working on, of course, and the unique challenges that they provide. I mean, you really can't look at them as simple small molecules, right? But certainly it comes to metabolism, but they are different than other modalities. So I think that's something that is an evolving area that they may have decided just simply not to include at this time.
From Paresh, does the new ICH guideline recommend evaluating pre-incubation, the TDI effect on all transporters?
No, they specifically call it OATPs, but they do reference some of the other papers that were published in the last several years that show an effect on most transporters. So that's why they say follow the literature. I think they were being a little cagey, actually saying they didn't want to make specific recommendations on the other transporters except for the OATPs, where that is quite mature. But I do think that time-dependent inhibition for the other transporters, and you can look at some of the papers. Salvo had a nice publication on that a few years back, and other references that can be looked at to decide how that should be approached.
And from Lashma Khan, if transaction efficiency varies for transporters, I think that's what transfection, probably efficiency varies for transporters, will IC50 value change?
That's an interesting question. So I guess it comes down, that would be in the equation, you'd be looking at enzyme concentration. And of course, we typically, for IC50 calculations, just ignore enzyme concentration. In this case, it would be the transporter concentration. So I don't know the answer to that off the top of my head. I would suspect that if there are large changes in transfection efficiency, there could be changes in IC50 values. So again, having a comparator would help in that case, and then also looking at, like a known substrate plus a known inhibitor, for instance, to validate that.
That sounds like an area for an ongoing conversation. And so yeah. we did not put Brian your email here, but I didn't. But Brian's email is b.ogilvie@bioivt.com. Did I get that right, Brian?
Yes.
Yes. So for all of you still on the line, if we're not getting to your questions or you want to continue the dialogue with Brian, again, it's b.ogilvie@bioivt.com. Let's see if we can get to a couple more here. Also from Juan, this is a follow-up. Follow-up to my question, are there equations that could, previous question, equations that could be used for hepatocyte and microsome unbound fraction?
Certainly there are for microsomal unbound fraction. I think I have actually seen in the literature some equations for unbound concentration in hepatocytes. I haven't paid that much attention to it, honestly. It's, in my mind, what is unbound concentration in hepatocytes? I think the best measure would be the KPUU. And if there are equations for that, I have not seen those. It wouldn't surprise me if there are, but there would probably be a little bit more intense than just the microsomal calculation.
Okay. And also a clarification from Grace. If induction time is less than 40 hours, does it require justification?
They do mention that you can use different time points, but yes, you have to provide justification, and that can come down to we have cytotoxicity at the longer time point, and we have to use lower time points. And there have been other cases where you actually see more induction at lower time points than you do at longer time points. And so if you have justification for that, then that would be a reason to use shorter time points.
So that actually brings us to the end of the questions and the end of the hour. We really appreciate the engagement from everyone and these great questions. Again, I would remind you, continue the dialogue with us and with Brian at bogilvie@bioivt.com. Also, please visit our website. You can receive notifications for future webinars and resources. Stay online for the survey. The survey also gives you an opportunity to sign up for additional webinars and other symposia that BioIVT is hosting. I think that does bring us to the end of this webinar, and again, a great engaging session. Thank you very much, Brian.
Thanks, everyone.
For your presentation and the thoughts. So thank you.